Good afternoon, everyone. I'm Will Rosenzweig. I'm the Dean and Executive Director of the Food Business School at the Culinary Institute of America. Happy to welcome you here to our FoodBiz Plus discussion this afternoon. And I'm going to just pause a few minutes because we've got quite a few people signed up, and just in consideration of their arrival, I'm going to begin in just a few minutes. So hang tight with us. You'll notice that we have a chat box where you can send your questions during the conversation, and Justin and I will do our best to weave them into the discussion, which will last for about 45 minutes. So I'll be back with you in just a minute or two, and we'll get started with our conversation this afternoon with Justin Siegel of UC Davis. Well, once again, welcome to FoodBiz Plus this afternoon. We're happy to have you with us today. This is our monthly series of conversations with thought leaders and pioneers in the world of food and health and science, environment, and business. And this week, I'm really grateful to have Justin Siegel, Professor Justin Siegel, with us from UC Davis. Justin is also a professor at UC Davis and on the faculty of the Food Business School. And if you so choose and you want to share this FoodBiz Plus webcast with your friends, you can tweet about it. Our handle, as you saw, is my handle is Idea Gardener, and the Food Business School is FoodBiz School, both on Twitter. And for those of you that are new to our sessions, the Food Business School is the Center for Executive and Graduate Education at the Culinary Institute of America. I'm based here in Northern California near our Napa campus, and we also have campuses in Hyde Park, New York, that's our main campus, and San Antonio and Singapore. The Food Business School is a bit of an innovation for the CIA, as we like to think of ourselves as being on-local. We're not only place-based, but a lot of our educational programs are delivered online, and I'll be telling you a little bit more about that in the future. Our mission at the Food Business School is to enable and empower our students to design, deliver, and lead transformative innovations that address the world's most pressing food systems challenges and its greatest business opportunities. So we really focus on applied education, on entrepreneurship and education and innovation, and we create programs that people can immediately put to work in their work or their education. And I'm happy to say that we're 1 year old now. We launched the school's first programs in June of 2015. And in the past year, we've run about 11 different programs online, short form in person, and then longer form in person. That one's called the Food Venture Lab. We've had about 350 people participate in all of our courses so far. And what's been interesting to me is 35% of all of our learners are from global locations. So already the reach of the online platform, as well as the appeal of coming to Napa to study at our home campus, is proving to be quite attractive. So the Food Biz Plus series is a way for us to share topics that are of great interest to us, and also have you get a chance to meet some of the faculty who are going to be teaching upcoming courses. Justin is leading a course this fall called Trends and Technologies Transforming the Food Business. And Justin brings a really remarkable background in science, in chemistry, and biochemistry, and biology. He runs the Siegel Lab at UC Davis. But one of the things that to me is most interesting about Justin's background is that he's also an entrepreneur and has been very much involved in translating science for the marketplace. And he's really in a hotbed of innovation. He's the new, the first kind of the premier director of the new Innovation Institute for Food and Health at UC Davis, which is quite an undertaking that was just founded in the last year. I had the good fortune of being part of the founding committee that helped to shape the constitution for that program, and I'm going to ask Justin to tell us a lot more about it. So without further ado, let me welcome Justin Siegel to the conversation. Justin, can you join us here online? You'll have to start your webcam. There you go. Okay.
All right.
You look like you're in a classroom. Are you?
I'm in my office.
You're in your office.
Yeah.
You've got a lot of equations written on the back wall.
Exactly.
Yeah.
There you go.
That's what theorem is that, Justin?
That's Graduate Student 101.
Okay.
How do you plan an experiment?
So, Justin, thanks for joining us today. What I'd like to do is learn a little bit about your background and how your work is now converging in the world of food. And I'd love for you to tell us about enzymes and proteins and the Institute for Food and Innovation. We can get started there. And then I'd like maybe for the second half of our conversation to focus a little bit on your course and how that's taking shape and to tell us a little bit about the topics and some of the activities that people are going to undertake. How's that sound for an outline?
Sounds great. Sounds really good.
So tell us about your background, Justin. You're relatively young member of the faculty there at UC Davis. We had the pleasure of having Bruce German, one of the senior members of the faculty, join FoodBiz Plus a couple of months ago, and Bruce focused a lot on the microbiome and his research with human nutrition and especially infant and prenatal nutrition. What do you focus on at UC Davis?
Yeah, thanks. So my focus is very complementary, and I work a lot with Bruce. So I've been here at UC Davis for about 4 years now. So I'm transitioning out of that new into kind of a steady state position here. And my major focus is on enzyme engineering and enzyme discovery. And so before I kind of go into that, I'll preface a little bit what enzymes are, just for the broader crowd.
And for my benefit too.
Yeah. So enzymes. So if you look around in nature, matter is transformed from one chemical composition to another. So sunlight and carbon dioxide, plants take that and turn those basic building blocks into everything that you see and everything we eat. Same with nitrogen and phosphate. So these basic elements are all built up, and through these stepwise processes of taking these basic building blocks, connecting them together, and then transforming their shapes until those shapes and structures impart the function you want as far as the molecular function. So the catalysts that enable that, because those transformations just don't happen on their own, the catalysts that enable that are called enzymes. They're proteins, except the difference between the proteins you're probably used to in like whey and milk and stuff like that. A lot of those proteins are there as a energy source, and they're stored, and they're there to provide literally protein to a body. These proteins you need in very small amounts because they perform these chemical transformations often 1,000,000 to 100,000,000 fold more effective than any known catalyst that we've made as a human today. And you can see from around us that everything we're doing now is developed through chemical catalysis. So a lot of what my lab does is try to think about how does nature have these amazing catalysts, and how can we utilize them to start harnessing their power and harnessing their ability to transform materials for what we would like today. And so I've, as Will kindly introduced, there's a lot of fundamental work. It's how do enzymes function? How do the structures and the shapes of these enzymes impart activity? And that can be applied to a lot of different areas. And so, as Will mentioned, that one of the areas that we're looking at pretty closely is food, even though we're also looking at energy as well as health. But food is kind of a central part there. So when you think about it, yep.
Let me ask you, how would you described a bit of the complexity of the enzyme, but if you were to describe its chemical or molecular composition, is it a wildly complex molecule, or is it rather simple? Where does it fit into the sort of the chain of chemistry, just so we can get a footing.
So enzymes have the beauty of being incredibly simple but incredibly complex. They're built out of 20 amino acids. You think pearls on a string. There's one of 20 pearls, and there's often about 500 of these pearls of the 500 amino acids on a string, all connected together. But they become complex because depending on the exact order and the exact number of positions you have, you get a different structure. So that pearl folds up into a different structure. It might look like this, or might look like this, or might look like that. It'll have a different structure. And that structure of the enzyme dictates the function. So the simplicity is that there's 20 building blocks. There's like 20 component parts. So there's 20 Lego bricks to play with. That's the simplicity. The complexity is that you can have 500 of them in a row. So that's 20 to the 500th possibilities. That's one of those numbers where there's more possibilities there than stars in the universe. So we have to come up with complex algorithms to think about how do you sample that space to predict which ones of those sequences will end up having that function you're interested in.
So when I bring that back to my kitchen and I think about an egg and frying an egg and adding a heat source to a protein in an egg, I'm creating a kind of a chemical and a molecular transformation. Are those the enzymes that are work at transforming that from a raw egg to a scrambled egg?
No, often what you're seeing there is proteins. And so what happens is these proteins, when they're at low concentrations and just being formed, they fold up into a specific shape. When you heat them up, they unfold. So that pearl, instead of having this very specific shape, it unfolds. And when it unfolds, the way these proteins, the physics behind them is the proteins fold up into a specific shape, and the inside has one molecular property and the outside has another. So it's a hydrophobic core and hydrophilic outside. So the inside really is repelled by water and the outside really likes water. And so it folds up to this nice stable structure. When you heat it up, it unfolds and exposes that hydrophobic part that doesn't like water. And so when you see the egg go from kind of a yellowish color to white, what's happened is you've unfolded all those proteins, and the parts, these long extended parts that don't like water, start packing up against each other into kind of an unstructured conglomerate, and then it turns white, and that's what you're seeing.
That's fantastic. So I think I understand that a lot of your work, because these enzymes are so simple and complex and offer such infinite variety, a lot of your work's computational, right? It's not physical. You're not working with frying pans and lab equipment. You're working with computers, right, to model the sequences of the proteins and enzymes.
Yeah, and so we do that often because when we think of functionality, so what happens when you eat a protein, for example, is it goes into your stomach and there's enzymes in your stomach, particularly pepsin is one that's well recognized, and in your lower intestine there's trypsin and chymotrypsin, and they recognize specific parts of these proteins and break them apart. And because you can think of a Pac-Man, maybe this protein has a structure like this and the enzyme has a structure like this, it recognizes very specifically your protein, whereas if the protein looked like this enzyme wouldn't recognize it. So these enzymes have a lot of specificity for the shape, and clearly this is an oversimplification of a shape. I'm trying to be very simple here. So this very this shape recognition, it dictates how these enzymes break down the proteins that you eat in the stomach, break down actually all the molecules that you eat when you eat something, and how they sometimes activate molecules or inactivate molecules. And so as far as new function goes, where our lab focuses is how do we harness the power of these enzymes, one of our fundamental goals within the lab is to make it so when you eat something, you get the exact nutritional profile you'd like to get from that food. So you can imagine having a hamburger pill or a pizza pill or a beer pill, and you would know when you pop that pill, it had the right composition of enzymes to break down those materials in a way that would be maximize the nutritional benefit for your body.
So are you proposing that everything you're going to do is going to end up as a pill, or are you looking?
It doesn't have to be a pill. I'm using a pill as an example.
I just wanted to clarify that because
Thank you. Yeah.
UC Davis, UC Davis can be accused of putting everything into a pill at times, so I didn't want to.
Yes.
But what you're
Yeah.
saying is you're trying to understand these interrelationships between external food source and our
Yeah.
ability to metabolize the nutritional value of certain foodstuffs in a way that potentially leads to personalized nutrition. Is that a goal of where your lab's work might lead?
Exactly. So for that, so you can eat culturally what you'd like to eat, and you can have your family traditional meals, but you get the nutritional benefits that you need, which is likely different than your parents and versus my daughters, versus your cousins, or some strangers. Everyone has a slightly different need depending on exactly their activity level, their genetics, and the environment that they live in.
So
And
when
so
you
so be able to come find those enzymes and those biological components, or build those biological components that help activate or modulate the food sources that we eat, we have to often look through nature or build on our own from first principles proteins or enzymes that actually are able to catalyze those unique functions that we weren't necessarily evolved to carry out. We were evolved as generalists, not specialists.
So if you start altering well, first of all, before I ask this question, I'm really interested in how do you actually interact or or deconstruct or reassemble an enzyme or a protein? How does a scientist actually go about taking apart something that you find in nature and putting it back together in a different way?
Well, so there's been advances in synthetic biology that have been truly phenomenal over the last, in genomics and synthetic biology, I should say, over the last 10 years, where we can now see the sequence of every protein that's out there in nature for pennies. It costs almost nothing to be able to see the protein sequence, the genes that encode the proteins that are out there in nature.
And do you have like a small tabletop sequencer in your office that you're just running things through, or what? Tell us a little bit of how that's working right now on a technical level.
Sure, at a technical level. So there is there's no tabletop there. I mean, people are coming out with tabletop sequencers right now. That is happening. It's questionable where they're going to be used and how they're going to be useful, because of the accuracy that they currently have. But there's a lot of central facilities. I actually happen to be sitting in one of these central facilities, so I do happen to have one down the hall, that have very expensive equipment, but equipment that people can send their samples to and they can obtain sequences for. And so it's outsourcing your sequencing, or it's almost cloud sequencing. You can imagine, as you send out your sequence and they sequence the date, they sequence the sample and they throw it up online, and you can access it there. So there's databases now online. Uniprot and NCBI are examples of these, where there's 60 million to 100 million sequences just sitting there, that people have sequenced at some point, that we don't know what most of those sequences are. We know they're a protein sequence, but we don't know what they are. And so where the modeling comes in is we can say, well, here's all these 60 million protein sequences that we have. And that was 60 million today. Tomorrow, next year, it'll be 120 to 150 million is the prediction. And it's growing, almost doubling, if not tripling, every year at this point. So there are millions and millions of sequences available. And so then you can take that sequence and you can run these computational algorithms based on all the great work that's been done in structural biology over the last 50 years to really predict what that structure is going to look like. And so all of a sudden you can take a known sequence and basic principles that we've learned about how proteins fold and what interactions are good, positive, and which interactions are negative, and predict what the structure is going to look like. And if you can predict a structure, often that can relate to function.
So by creating a reaction, if you will, you can reshape the structure in a way that the way you intend. Is that
Exactly. So when now the trick, exactly like you said. So the trick is, once you have a structure that you'd like, that you think has that sequence, how do you actually get that in the lab? And so now at this point, you can literally type in onto the computer the sequence of the protein you want and the organism you want to grow it in. We often use standard lab strain, generally regarded as safe organisms, to obtain genes that encode that protein that we've modeled. And it takes a whole 10 minutes to type it into the computer and hit order. And about 5 days later, that gene shows up. So within a week or two, you can go from a picture on a computer to something physically in hand for within a roughly 2 week period for $100 to $200.
And is that living gene? I mean, is it in a, how does it arrive? I haven't seen one. So
They arrive in a little, it's very anticlimactic.
yeah.
They arrive in a test tube
Uh-huh.
that you can't see anything in,
Right.
and you put some clear water in there.
You
You
take their
still
word
can't
for
see
it.
anything.
You take their word
Yeah,
for
and it's
it that
there
you got
though.
what you ordered. Okay, so
Yeah.
give me an application. Give me sort of something, a basic example of how you might think about applying this in the real world. Where might this lead? Am I looking for, like, let me give you an example. Just 2 weeks ago, last month, we had Lisa Mosconi on FoodBiz Plus, and she's a neuroscientist at NYU, and she's studying the relationship between food and lifestyle and Alzheimer's and dementia.
Hmm.
And she's developing she's doing a lot of MRI research to understand how different diets manifest in different cognitive states of decline. At your level of research, at this biochemical level, would you start being able to isolate foods and proteins that might play a role in the implications of disease? Or tell us more about how you see applying this in the years ahead.
Sure, and I think you probably know this example that I'll give pretty well. I think that the cleanest example, it can get more complex, but the cleanest example is something that I've actually started a company on, and that is moving forward, and we're hoping to start human trials later this year. Is for celiac disease. It's not gluten intolerance, but it's essentially if you have gluten, you have an autoimmune response, and there's a whole series of different symptoms that it could arise and that you might see from that. But roughly 1% of the population has celiac disease. And so if they are exposed to gluten, in that if they eat it, it's fine if they touch it, but if they eat it, it triggers that response. And some people have to go to the hospital, some people have just extreme discomfort. It's hard to predict with an autoimmune response in these cases. And so where you can imagine, where we're actually applying this technology, is saying that if you identify, and in this case, there was some great work done by Chen Kosla at Stanford and many others around the country at this point. They identified the exact regions of gluten that trigger the response. And so we were able to say, if we know that region of gluten that triggers the response, we can come up with an enzyme and cut it and break it. And so at that point, there was no enzyme necessarily naturally evolved or out there in nature that was able to perform that function, because we're eating wheat at a level that we were never evolved to eat it at. And so asking for an enzyme to prevent this autoimmune response for a specific product that's new relatively within our human diets, it just wasn't part of our evolution. And it would have been naturally selected out for, but it's no longer being naturally selected out for because
So
we
is
have
your
these
thesis
modern
then that
medicines.
you'll populate a person's gut or their microbiome with a new enzyme that
So
will,
in this case, we're avoiding the microbiome. In this case, it's a pill, and that's why I was using the pill examples earlier. Is you would take a pill that has this protein or this enzyme, because it's a protein with a catalytic function, that breaks apart the regions of gluten that trigger the disease. So someone with celiac disease can eat a hamburger or a bowl of pasta, or just go out to dinner at a family's house and not have to worry about whether there was flour used in the kitchen or not, or they could actually eat the meal. So it's a devastating disease for those that have it, but we're hoping that this will actually be able to help it. And this is a first example of where we think that this will be able to be utilized.
fascinating.
The microbiome is an interesting question, and I'm sure Bruce talked about it a bit before. Is there's always a an interplay between how, if you want to really modulate the microbiome, and how do you do it in a way that maybe isn't permanent, um,
Hmm.
or maybe you do want it to be done permanently. And so we're just starting to learn about the microbiome. So there's a little bit of a danger as far as just we don't understand the consequences yet of manipulating the microbiome. Whereas we do understand the consequences of introducing a protein supplement into your food source or a digestive aid into your food source. That's been done for a long, long time now. And so that's where the idea of starting with pills, where you're adding a digestive aid essentially. In this case, we're going for a drug, so it's medically diagnosed and there's been clinical trials done behind it, into the food system. It's pretty well established. The next frontier will be manipulating the microbiome, but there's a lot of fundamental studies that need to be done first before I think anyone will be really comfortable manipulating the microbiome, unless there's a real case where it needs to be done. And so C. diff and some of these diseases where you see uh, 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 that microbiome transplants are required and actually saving people, are very exciting and suggesting that it is possible and that this might be a good thing. But the granularity that's a complete swap out as opposed to fine
Hmm.
tuning it.
So, Justin, talk to us a little bit about the cross-disciplinary collaboration and interaction that's going on with scientists now to get at this very complex, um, human biology, um, food, health. What does a team look like of expertise that is able to not only develop the core science but think about bringing it to market? What does an entrepreneurial team look like for, say, the company you just described?
Sure, and I think so. The company I just described is more of a drug company there, so it's a pretty clear cut team as people experienced in the drug industry with how do you have design clinical trials, how do you develop a business plan, how do you have the science for producing that product, the producing that protein of interest, so a fermentation of some sort, and then the science of how do you actually have a compound that does what you want, an enzyme that does what you want. So it's kind of like four cornerstones, I would say, in that sense. But that's for a very smallly defined product. I think the bigger question is, and this is what we're starting to do in the institute, and I don't know if you want me to transition
Yeah,
a little bit
please. That'd
to
be great.
that, um, is in a food system is how do you affect that, and how do you transform the food system, and what does a team look like there? And that's something we're just starting to explore at the Innovation Institute for Food and Health, which is what we will have mentioned we started about a year ago at this point, and will be becoming much more public, I think, over this next year is our plan at least. Uh, and that is bringing together academic, it's multi-sector and multidiscipline. Uh, because unlike in the drug world where people can control it end to end, or in the computational world where you can control it end to end, there's no such thing as controlling something in the food system end to end. There's no one company that owns all the fertilizer companies and all the growers, owns all the processors, owns all the distributors, owns all the chefs, and owns all the waste supply. There's no one that owns that entire system and can control that entire system. Whereas in my Apple and my
Goodness.
Mac here, there's many supplies and it comes as it is, and I get to use it as it is. So you have that end-to-end control. So when you're trying to have systemic change where there's many, many different stakeholders across this complex system, each one with different incentive structures, which they each should have their own incentive structures, but it's how do you align those incentive structures? So how do you get academics that are trying to understand what's happening in our food system, working with industry who are trying to both affect and profit from that and make a living from that, the government sectors that are regulating that to make sure it's safe and done in a sustainable, longer vision than the profits can drive on its own way, and the civil sectors to make sure people are getting what they want, and it's done in an equitable manner. How do you get all those four sectors working together?
Well,
And
and
so we're
not to
really trying to.
mention, not to mention the fancy of the eaters, all 270 million of them in the U.S. and coming soon to a planet near you, 9 billion on the
Yeah,
planet, right, by 2050 with, as Eve Paul told us, a couple of months ago. She showed us really the power of the millennial cohort as a driver of new trends and preferences and tastes in food. So I always think that, of course, I think from a kind of a consumer-facing perspective, those expectations weigh heavily on perhaps they should weigh heavily on the activities and mindsets of the scientists, particularly as we're wading into areas of genetically modified organisms becoming a greater part of our food system. Maybe we could talk a little bit about that and how
Sure.
how that fits in this kind of creative tension between people having this pastoral, pristine idea of the natural world and our natural food system and our local and organic food system, and now our increasingly sort of science-based, biology-based understandings of the food system. Talk to us a little bit about that.
Sure, and I think the first thing I'd say there is so there's two things I'd say there. The first thing is that I definitely agree with Eve. Like, food is culture, and if anyone thinks that they're going to disrupt culture in a single lifetime, they're probably highly mistaken. Like, so you have to think about how do you adjust food system within the culture and the values that we hold today.
Yes.
And that's really, really important, and something that you don't have to ascribe to in the drug sector or the energy sector. And so it's something that's very interesting that all of a sudden the anthropology and the society and our values really start making a direct impact on the products and how they move through market and how successful they are in the market. So it's a totally unexplored area as far as the startup space. And so as far as
it's
GMOs,
a different mindset, right? It's really, like you say, different from if you've got people in your lab that are working on energy problems, just they can't apply that same mindset necessarily to food. They can apply some of their technical knowledge, but they really have to inhabit a different mindset about solving a problem.
exactly. And it's how do we adapt these tools to what people need and want, as opposed to just what they need. And so it's a fundamentally different mindset that a lot of scientists have a hard time with, because a lot of us come from the energy or the medical sectors where that mindset does not exist. It's we know what is needed and we will develop what's needed, as opposed to necessarily what's needed and how do you make sure it's also wanted. Because it's always a choice now when you get to food, as opposed to a need in this case, for now.
Right. I mean, it's become clear to people that they don't want pesticides in their food that have implications for disease. So, I mean, this is...
And that's an interesting point to transition to the GMOs, because I think a lot of times, as you were saying, is we think about you have organic and pristine and naturally derived heirloom products, and then you have GMOs that are engineered and biology driven. And I don't think they actually have to be separated in that way. You can have GMOs, genetically modified organisms, that are pristine and that are actually more organic and more sustainable and require less chemical inputs than any natural source. So it's really dangerous to combine GMOs with pesticide usage or fertilizer usage or water usage. Now it is done that way because unfortunately the first round of GMOs really focused on that. They focused on what's best for the industry and not necessarily what's best for society and the overall environment.
You're making my point for me. You're reinforcing that potentially that group of scientists were not thinking with this systems mindset on. They were trying to solve an industrial problem, which they did quite effectively, without fully understanding the implications of the systematic consequences of their work, right?
Yeah, absolutely. But the beauty is GMOs have the power to make it so maybe we don't need any pesticides and we can reduce water usage by tenfold and We don't need any fertilizers because there's enough in the natural environment. You can imagine GMOs, and not really just imagine, people are very actively working on this. There's a colleague right above me right here at Davis that has made it so you can have a significantly less water input into several crops, and it's fine. So it's how do you reduce the environmental impact and reduce the need for all these different inputs, both natural and chemical, or synthetic, I should say. These are all chemicals in the end. uh, but make it so it actually is more organic and more natural than you could ever achieve through nature alone.
This collides also with another topic that's near and dear to food business school learners, which is really how is this science communicated to the public um, in a world where transparency is a very important value. You know, last 2 weeks ago, we had Gary Hirschberg, who's the co-founding CEO of Stonyfield Farms, a great proponent of organic food production, but also now the chairman of the Just Label It campaign. And he came and spoke to our class. He was very clear, like you have just been, about that this is not about being anti-GMO. This is merely creating the rules that that business should declare, should be compelled to say what is in their product or where it comes from,
Yep.
and just insisting on more transparent communication with the consumer so that consumers can at least know what it is they're buying.
Yeah, that's absolutely, and they should understand the overall impact of what they're buying on the environment and the system. And so so another very complicating factor is that we really don't understand the food that we eat today or how those factors affect us. There has never been a single study that equates to a phase 3 clinical trial like we do for drugs for any single item in the food chain.
It's fascinating.
Not a single nutrient, not a single additive, nothing. So we know more about every drug we put in our body than any nutrient that we put in our body, which is a very weird thing to think about when we use drugs on a rare occasion, hopefully, and when it's needed, as opposed to something that we need every single day and actually dictates the need for those drugs down the road.
And Justin, that's so fascinating. Do you think that's driven by intellectual property protection and investment? I mean, there's hundreds and hundreds of millions of dollars invested in drugs because people can protect them and then monetize that investment in the marketplace. But with food, it's a very different landscape in terms of that type of business opportunity. Is that right?
That's exactly so. There's two aspects, I think. So the first is how do you monetize that? If you spent $100 million, which is, I'm not throwing that number out. That's actually, if you want to do a phase 3 clinical trial, $100 million is the ballpark you'd be expecting to spend if you have 10,000 to 150,000 people with multiple arms, meaning a control group, different levels of doses in different areas to try to understand how your environment affects that nutrient or that compound that you're looking at, plus blood work and check-in and the doctors and everything. It's about $100 million. And so how do you make back that $100 million if you do that study on a nutrient that's found all over the place and then everyone can profit from that, as opposed to holding a patent and holding the rights to something for 20 years or 15 years after all is said and done with the clinical trials and the normalization from the FDA. It's coupled with that is how do you protect it? And the other factor is that drugs have an insurance agency and can be sold for very high rates. So it's thousands of dollars per pill or per item in that sense, as opposed to $1 or 50 cents. Even if you only pay $20, they might make a lot of money on that drug still, from an insurance company's point of view. The insurance company is helping supplement that. So there's a much higher profit margin, and you can protect it. And so how do you do that in the food system is a real challenging question. And it's likely one of the reasons that it hasn't been done. And this is actually where I think one of the areas that the government needs to step in, because it is a basic human right to understand what we're eating.
So at the institute that you're growing there at Davis now, the word food for health is in the title. It's not drugs for health; it's foods for health. So,
Hmm.
um, I'd love to hear a little bit more about that, and I'd also think we should probably transition to talk a little bit about your course. And maybe just as a prerequisite to that, I'd like to say one of the things that's so great and exciting about FBS, Food Business School, to me is that we get talented people like Justin, who, for the most part, are sequestered in small courses at UC Davis, and we get to open up your knowledge and your network and your experience and your view to a much wider audience, because the online platform that your course will be delivered on will be open to
Yeah.
anyone who wants to apply. So maybe tell us just a little bit more about the IIFH initiative, and then talk a little bit more about your course, and we'll wrap up our conversation today.
Sure, and I think they're almost one and the same. The IFH initiative is what I was getting to earlier, is thinking about multi-sector, multi-discipline groups, and how do you set a stage so everyone has similar values and goals, and understanding we're not going to change incentives, but how do we understand each stakeholder's incentive structure such that we can build projects and we can build programs around that as a team, that where everyone wins, where the industrial groups can make a profit through developing a product, where society gets the foods that they want and need, where the academics can do the fundamental research to help define those foods that people what they want and need, uh, and through the governments to make sure that these are wrapped in a way that both ensures safety as well as ensures the uh, products are put together in a sustainable way,
Right, and
and
that's all
the
taking
policies
place
match
in a
that.
context of really rapid uh, kind of reimagining of how food and human health and planetary sustainability all interact on a
Yeah,
on both a sort of macro systems level and a molecular level, inside of us and in the soil, right? So that's the context, is trying to create environments both outside that we live in and inhabit, and environments that exist inside of us that ultimately lead to flourish, to enable us to flourish for the long term and in a
absolutely.
fair and equitable manner. It's an enormously complex and huge responsibility.
Yeah, no, it's an exciting area. And in addition to understanding the fundamental, how do you have these systems, work together in a way that's holistic and good for the environment and good for people, and able to provide what's needed for people. It's how do you even get a large multi-billion-dollar corporation to work with an academic and to work with government and to work with society in a way that everyone feels is productive, and everyone feels that understands and is done in a transparent way, as at least transparent as possible, while maintaining that incentive structure to actually have. In the food business school, we're in a capitalist environment. It's only done and only sustainable if it's not relying on donations and not relying on someone's goodwill. And so, how do you do that in a sustainable way where there's actually some kind of profit driven? It doesn't have to be a huge profit, but there's some kind of profit driven model behind it to make sure that it's a sustainable solution.
Mhm. So just tell us a little bit, tell us a minute or two about your course. It's coming up this fall. It's going to be, is it 5 weeks in length or 6 weeks?
Yeah, it's 5 weeks,
Yeah.
and it'll kind of go through. It'll start off, the first week will be a broad overview of the food system to try to really define the food system and provide a good context of what the food system, the different aspects of the food system are, and general technologies and trends that are happening within the food system, ranging from using genetic testing and DNA sequencing for food safety, to robotics for harvesting, to enzyme engineering for manipulating the nutrient profiles we eat. And then each subsequent week, we'll kind of go through a more focused section of the food system, whether it's production, processing, distribution, or consumption, and then subsequently waste. And clearly some of those will get mixed together a little bit. What are some major, we'll focus there instead of the technologies themselves, but what are the major problems and the opportunities that exist within each of those sectors that really people that I would love the food business school to, and participants of the food business school to start seeing that there's opportunities here of both a business model being able to capture that gap that exists.
So part of the class, you'll be challenging the students to recognize and define opportunities where these technologies might be coming to market and potentially hypothesize how they might come to market, right? In
exactly.
some kind of
or technologies that are needed that need to be developed.
Okay.
Yeah, it doesn't necessarily, there's a lot of technologies that you can imagine developing rapidly if you understood the problem behind it. Or I have a feeling there'll actually be quite a few people in the class that once they become knowledgeable about a particular problem in the food system, they'll realize that their technology might have a solution towards that problem.
Well, we're really excited to have you teaching with us. I know that one of the 3 primary goals of the Siegel Lab at UC Davis is to further the innovation in teaching. So
Yeah.
I really appreciate that you've been one of the first people to jump aboard here and help us invent a new course. And we're really looking forward to that being offered this fall. And a couple of our listeners today have sent in questions to us, and we'll get back to you folks with specific answers to the questions that you posed that we haven't been able to cover in the conversation today. So Justin, thanks again for coming. Justin Siegel, professor at UC Davis. See you soon, Justin. Take care.
Thanks so much, Ron. I'm looking forward to the course. I think it's going to be a phenomenal opportunity for me and for the students, hopefully.
Great. Thanks. Take care. I'm going to share with our learners now just a sampling of some of our upcoming courses. This is all available on our website, which is foodbusinessschool.org. You can look and see tech trends and policies. That's Justin's course coming up. We've got Essentials of Nutrition and Sustainability, which is going to be a great course with Bruce German and Tom Tomich. Eat, Think, Design. This is the first time we're going to be offering this course. It's very exciting. It's really about design thinking and opportunity recognition in food. It's taught by Professor Jaspal Sandhu, who's been teaching this course at the UC Berkeley School of Public Health in the master's program there for a number of years. It's a fabulous course. I've had the pleasure of working with some of the students that go through his course, and people really learn about how to kind of spot the white space in a sector and develop and define an opportunity and then develop a hypothesis for a solution and then test that solution. That's just going to be very exciting. So Tech Trends and Policies, Essentials in Nutrition and Sustainability, and Eat, Think, Design. These are all 5-week online courses. They're very affordable. They're very high value. They're intended to be at the graduate level. So some understanding of the food system is anticipated, but if you're an undergrad, you're certainly welcome to take them as well. And then we've also got this fall From Concept to Shelf. This is one of our 2-day innovation intensives. This is a course that's intended for people that are either starting a packaged food product company or people that already have a company that are contemplating their next product launch or extension. And it's a really fun and action-packed strategy session that I actually co-teach this with Chris Cornyn. Chris is the Chief Innovation Officer at Revolution Foods, and before that had his own innovation agency in food. Really, truly one of the most creative, talented people that I've had the pleasure to work with. You'll learn a lot of tools for innovation, of how to lead your team, how to generate lots of ideas, how to filter those ideas and test them rapidly, so that you can develop the really comprehensive, holistic new product strategy and form. So we teach rapid prototyping, we focus on commercialization, and of course, when you take any one of our food business school courses, you actually get credits for our mentor network. This is your opportunity to work with, we have 40 really just accomplished people on our mentor network right now who you can interact with like this online, live, in personalized one-to-one sessions. So anytime you take a food business school course, you get credits for the mentor network. And if you have questions and you want to ask anything more, you can talk to Nancy, who heads up our student experience department, and her email is admissions@foodbusinessschool.org. And you can also benefit from a 15% discount off your first course for spending this time with us. We certainly appreciate your interest and attention and your support. We're really a fledgling startup ourselves. We're a nonprofit educational institution. Anything you can do to share what we're doing with your friends and colleagues and put in a good word for us, we greatly appreciate it. So thanks for being with us today, and thanks for being part of Food Biz Plus. Good night.